One of the most powerful techniques a cytogeneticist has is a technique called fluorescence in situ hybridization, or FISH. And what FISH allows cytogeneticists to do is to assess the presence of very small deletions, inversions, translocations, and other cytogenetic rearrangements between chromosomes. So in fluorescence in situ hybridization, as usual, we start off with a glass slide that contains a metaphase chromosome spread. In this case, I'm just going to look at one chromosome. Two sister chromatids next to each other on a slide. And if we zoom in, we would see that each of those sister chromatids, as we already know, is a double-stranded piece of DNA, with the two strands held together by hydrogen bonds. Through the process of denaturation, those strands can be separated, melted, so that the individual letters of DNA are now separated. So we might have TAG on one strand and CTA on the other strand. At this point, a cytogeneticist applies a DNA probe that has been fluorescently labeled to the metaphase chromosome spread. Such a probe is usually a linear piece of DNA that is also single-stranded and also has sequence homology, that is, it has a similar sequence to one or both of the strands of the chromosome. And as I mentioned, one of these nucleotides, sometimes G, is labeled with a fluorescent chemical, so it will indicate that this G has a red fluorescent molecule attached to it. And this, again, is the probe. Single-stranded DNA that's fluorescent. When the probe is annealed to these single-stranded sister chromatid bits that are stuck to a glass slide, again, this one has the sequence ATC, that probe now can anneal because of sequence similarity. T goes with A goes with T, G goes with C, and that G is fluorescent red. So if we use a fluorescent microscope now and we look at this microscope slide, we would see that on this very end of the chromosome shown that there would be a red fluorescent dot. And that fluorescent dot basically tells us visually where on the chromosome the sequence TAG, the probe sequence, exists. Now, probes in reality are never 3 base pairs long. They're often the sizes of genes or entire sections of chromosomes, 100 to 200,000 base pairs long. And again, this technique is used just to look for structural differences between normal and perhaps abnormal chromosomes. Now, I should point out that because the chromosome on this slide, the two pairs of sister chromatids that is, are identical because they're sister chromatids, you never see just one red spot on one chromosome. You expect to see two fluorescent signals at the same position on both sister chromatids, again because they're identical. So if one sister chromosome has the TAG sequence near the telomere, so will the other sister chromatid. And here's an example of what fluorescence in situ hybridization results look like. In this case, a purple fluorescent DNA probe has been annealed to this metaphase chromosome spread, and it is indicating for us the positions of that DNA sequence in the entire genome. We see 2 dots on each pair of sister chromatids, on each dyad, and that is, again, as I just described, because those 2 sister chromatids are identical. The reason that there are 2 dyads that are labeled is because those are the homologous chromosomes. Remember, this is metaphase, so we expect to see 2 chromosomes, or what we call 2 chromosomes, 2 pairs of sister chromatids that are homologous. So this is one way, like chromosome banding, that scientists can identify which pair of chromosomes in a metaphase chromosome spread are homologous chromosomes. In this case, because the 2 homologous pairs of sister chromatids share this fluorescence in situ hybridization signal. That's an indication that they have homology or identity, that they're related.